Hello, everyone, and welcome to our Florida Native Plant Society October Lunch and Learn. I'm pleased to be here with my colleague, Lily Anderson-Messick, and our presenter, Romina Gazis Saragina, PhD, of the IFAS Tropical Research Center, correct?
Research and Education Center.
Research and Education Center in Homestead, Florida. So I'm Valerie Anderson. Obviously, we are the Florida Native Plant Society. Your membership in Florida Native Plant Society supports these programs, and all of our work we do to conserve, preserve, and restore the native plants and native plant communities of Florida. So, would you like to introduce yourself?
Yes, and I'm also going to try to share my screen. Can you guys see it?
Yes.
Yes, perfect. All right. And you can see the full screen, right? Hopefully. Yeah, okay. Yeah, so thank you for the invitation. My name is Romina Gazis. I'm an associate professor in the Department of Plant Pathology, but I am based at the Tropical Research and Education Center. Sometimes people call it TREC or TREC. As you know, UF has several research centers across the state, and we are located to the southern most far away from main campus. So we are located in Homestead. I'm also an extension specialist with the university and the director of the plant clinic based at the center. And so a little bit about me. Again, I'm an extension state specialist for plant pathology of tropical and subtropical crops, including ornamentals, either in production or in the landscape, of tropical fruits and of natural ecosystems. So even though I don't work a lot with natural ecosystems, I'm really interested in this. And of course, if there is an outbreak or something that is going on with the plant health of these natural ecosystems, I will be the one to call. I have a 60% extension and 40% research. And I have multiple research projects going on tropical fruits. I am really a mycologist by training. So a lot of my research before this position was on fungal diversity, on the role of fungi in ecosystems, either as mutualists or as parasites. And so I'm really interested in entomopathogens and just how fungi really has a really important role in carbon cycle and all these recycling of materials in the forests. I also work a lot with endophytes before, which are fungi that can live within plants without causing any symptoms of disease. Sometimes these endophytes can change into bad fungi, and sometimes actually they are helping the plant cope with abiotic stressors. So if you look at my research previously, I did a lot of work on fungal ecology, fungal biology, and their interaction with plants and insects. And so again, we are located at Homestead. It's really at the entrance of the Florida Keys, and you have the Everglades on one side, and then you have Biscayne National Park on the other side. We have a lot of nature around our areas, but also a lot of agriculture. And we have climate and a weather that is very different actually to the rest of Florida. So sometimes we don't share a lot of the plants, but we do share the plants with the Caribbean and with the cultivated ones. We share it with Asia and other tropical places. I know there are several chapters in this organization, and there is one in Dade. So that chapter will be the closest to where we are. The Tropical Research and Education Center, if you haven't heard about us, is an amazing center. We do a lot of research in several aspects of tropical and subtropical ecosystems, either agricultural or natural. And we also have faculty that works with water sciences to preserve the integrity of the watershed and water-associated ecosystems, which you know we are surrounded by these kind of places. We also have a germplasm collection for avocado, mango, and other fruit crops. And there are 18 faculty members. Again, multidisciplinary. There are ag economists, soil scientists, people doing precision agriculture, people doing basic fundamental science in breeding, in plant health. So we are a bunch of people interested in the health of ecosystems. So this is an aerial photo of where we located. As I mentioned before, we are surrounded by farms, and these are very small plots. So these are small holders that grow sometimes a mix of cultivars and a mix of crops. And we are also urbanized, and we have some patches of natural lands like pine rocklands. They're very fragmented and they're kind of intermixed within the county. And we are ground zero, as most of Florida, for exotic species, but more importantly in our area, because we have the nursery industry, which accidentally move a lot of these invasive species, and we are a mix of ethnicities, and we all want to bring something from home. And so that has also been something that we have suffered from, people bringing plants from abroad without the required certifications and permits. So we really see a lot of these invasive species that move from the agriculture landscape into the urban landscape and then into the nature parts of the county. This is my lab. I call it the Research Lab and Tropical Plant Clinic, but it's really the Gazis lab. And if you want to follow us, we post a lot. Actually, I am the one managing the Twitter account, and we post mostly, like almost every day, about what we do in terms of research, of extension, outreach. So if you want to know what's going on with plant health of most of southern Florida, please follow us. And if you want to see some of the previous publications from my past life as a mycologist, you can also access those through Google Scholar. Again, we are interested in topics related to plant health, in horticultural crops and natural ecosystem. We like everything tropical. So if you see something like an outbreak in in one of these natural areas, please give us a call. We can definitely look into it. So my program goes from fundamental to applied. Sometimes I do very basic research, and sometimes I do very applied research. Nowadays, there are so many pressing problems with pests and diseases that I work a lot on applied research topics, right? Because we need to find solutions. We are a land-grant university, so we are stakeholder driven, and we need to offer solutions that can be implemented. And so there are many tropical fruits, and so there are many diseases affecting these tropical fruits. And I know that I'm talking to a crowd that may not appreciate symptomatic fruits, but this is what brings me joy. It's just beautiful to my eyes, and I get different reactions depending who I'm talking to. But this is what I work for, to allow or to facilitate growers grow fruit that do not look like this. So that's the idea, right? So they don't lose money, so they don't have to invest in resources, including labor, for scouts, for removal of plants, and also pesticides. So my job is to diminish and reduce the use of pesticides and use it in a way that is judicious and effective. And so these applied projects will depend on the funding. So it depends who gives me the money and how much money there is and the needs of the stakeholders. I change my research program towards more applied and depending on which crops. Avocado, dragon fruit, passion fruit, those are crops that are emerging. And so the state is putting some money to study the potential diseases that may impede the growth of the industry and the well-being of the farmers. And so it will depend what I work on based on funding. I also work on, again, my real passion is natural ecosystems and the role of fungi in these ecosystems in relation to plant health. So I do some of this research through my students, and this was Elena. She now works for the USDA Forest Service, but she did her PhD in my lab, and she looked at ectomycorrhizae in the pine rocklands. So I'm sure you know everything about the pine rocklands, and but the fungi was really understudied. And we know that they are very important for pine seedling establishment and survival. And so she looked into characterizing this community of ECM fungi and did a lot of greenhouse testing to show who are the players that are important in this environment. And so she did a lot of greenhouse testing, a lot of characterization. We were trying different commercial products to see if we can replace some of the native ECM with commercially available ones, especially for people that are working on reforestation or, all these campaigns that give away pines and then they die. So we want people to be excited about planting pines in historically places where pine rocklands were present. So this inoculum is present there, but when you move away from these areas, the ECMs are not there anymore, these fungi. So we were testing different commercially available formulations, and you can read everything about this in those QR codes. It has been published. We have one under review that is about one publication under review, which touches on the effect of sea level rise and salinization of the soils and how that affects the fungi that are mutualist to these pines. So, multi-layer relationships within players in these ecosystems. I also work with, again, with endophytes, and we were isolating endophytes from within pine trees in these pine rocklands. And we were able to recover some very cool fungi, especially in the group named Tolipocladium. It's a genus that I'm really interested in because it has relatives that produce metabolites that are quite strong, and some of them are mycoparasites and entomopathogens. So I see the application of these strains. We recently described 2 new species for science from these habitats, which, we are all trying to give value to these habitats. And one way to give value is to recognize them as source or reservoir of new species, right? And so we described 2 species, and one of them we named in honor of Trek. So it's named Tolipocladium trekensis. So I just find that endearing because I work here, right? My colleagues, they think it's cool, but they are not as obsessed as I am. But today I am here to talk about avocado. And as you know, it's a world obsession, right? People are increasing their consumption enormously of avocado. There was a huge campaign decades ago, maybe a couple of decades ago, to familiarize American families with the fruit. Because if you think about it's, dark and kind of rugose, and it doesn't look like, like beautiful, like a dragon fruit or, something colorful. And so it was really difficult actually for the industry to break that misconception that this is not a nice fruit. And they invested tons of money actually in education towards the benefits of eating avocado. But now it's not necessary, right? People eat avocado toast everywhere, guacamole. So you have people from Russia, from Nordic countries eating this tropical fruit. And for me, that's amazing. I was raised with this fruit. I'm from Peru. So it's normal. right? But for other people, not so much. And now you have a lot of other countries eating this fruit. So because of this demand, now the production has expanded quite a bit. And there's a lot of countries now that are competing to get into the European and US market. Still, Mexico is number one, but actually my country, Peru, is number two, way far away from Mexico, but still we have a really good production. And if you are Floridian, you know that we grow tropical avocados, which are huge avocados of the West Indies and West Indies Guatemalan heritage. And you can feed a family with one avocado, right? Sometimes we call them slimcado because they have less fat, though that campaign really hasn't caught up in the market. But you can call them tropical avocados, green skin avocados, and so on. And so avocado is affected by many diseases. Some of them affect the fruit pre-harvest. Some of them affect the fruit post-harvest. I'm sure you have experienced buying avocado and then putting it in the counter, and then in 2 days, they're covered with black spots and sometimes with some spore masses. So those are post-harvest diseases. Some diseases can cause abortion of the fruit. Some of them branch dieback, some of them root rot, and some of them can kill the plants, the young plantings. But none is as devastating as Laurel Wilt. You can see here aerial photos of Homestead, and you can see those patches of bare soil. That's where avocado trees were before, and those were killed from Laurel Wilt. So since it was first detected in South Florida in 2012, sorry, it has killed at least 300,000 avocados, and a lot of the backyard avocados as well. So it's a very important disease for us. And it's a disease complex. So it's caused by a fungus named Harringtonia lauricola. It's in the Ophiostomatalis group. Sometimes they're called blue stain fungi. And this is a vascular disease. It's a vascular pathogen. And it is vectored by a beetle, the redbay ambrosia beetle, that has these structures where the fungus is kept alive until the beetle moves the fungus from one tree to another one. One particularity of this beetle is that it seems to infest healthy trees, which is very different than the regular bark and ambrosia beetles, which ends up killing the tree after it has been in decline, right? So these other, I would say most of the ambrosia and bark beetles are attracted to dying trees. So we don't care much, right? Because they are kind of finishing up those trees that were on their way out anyways. But in this case, this particular beetle affects healthy trees. And so the female is the one that moves the fungus from tree to tree, and it makes galleries, and then it cultivates the fungus. So the adults and the babies, so the larvae will feed on this fungus exclusively. So this is the reason why you cannot, or we cannot use insecticides, systemic insecticides, in comparison to other pest diseases, pest problems, because the beetle does not eat the wood, it eats the fungus. So doesn't care if it's full of pesticides. And so the female, it's already fertilized when it leaves the galleries. So it's already ready to put eggs. And if it's not, it can put male eggs and then mate with those sons. And so it has this haplodiploidy inbreeding system, which makes the disease complex more complicated, because that means that one female is able to start an outbreak. Now, this is a very virulent pathogen, especially because, not because it secretes enzymes or toxins, but because it can multiply really, really quick as soon as it gets into the tree. So this is the reason why it's so virulent. The good thing is that the redbay ambrosia beetle does not like avocado. It does not like avocado. It only goes there when it's pushed to go there. Push, I mean, there is no other host around, so the beetle is going to have to go there. But it doesn't like it, which is good. However, in our region, in South Florida, and actually this is a phenomenon that has been now documented in other states like Tennessee, the fungus has migrated to other locally adapted beetle populations. So there are other ambrosia beetles in our area that are invasive or naturalized or native. And those populations have been infected with the pathogen. So now they have acquired this nutrient symbiont from the redbay ambrosia beetle, and the fungus now lives as symbiont of these other beetles. So now we don't have only one beetle, but we have almost 9 beetles that are moving the fungus around. So that's bad. So this disease has caused tremendous impact in natural ecosystems. As you know, it has killed more than 500 million redbays. It has pushed to the almost extinction some of these very unique, fragmented, endemic, small population lauraceae. And you know, it's a big problem. It has been found in the Everglades, and if you drive around, you can pinpoint the wilted trees that are very easy to see, kind of like flagging. Here are some photos of either a redbay or a swampbay. I think it's a swampbay, actually. I cannot remember. I was trying to remember this morning. And again, I'm not a good botanist. I'm just a good pathologist. And so this was these photos were taken near Gainesville. And you can see the wilting, the dieback, the streaking of the vascular system. You can also see where the beetles infested the tree. You can see those dark lesions. And you can see in the last photo to your right how the xylem has collapsed. And so you see the staining on the xylem, even very deep in the tree, which is very interesting because, you know, as the tree grows, a new xylem is created, new rings, and then the ones in the inside kind of become non-functional, right? And become hardwood. But this pathogen can really colonize even deep into the wood. And here are some photos of sassafras, one of my favorite trees. It's just gorgeous, gorgeous foliage. It's just beautiful. And it's a very important tree for the Southeast USA and also the eastern part of US. You know, it goes up all the way to Canada, actually, the distribution of this tree. And you can see here the same symptoms, right? This already advanced because it has defoliated, but also these tree defoliates. The other ones are evergreens, right? And so you can see that it's completely defoliated while the other ones have still canopy. And you can see the streaking of the vascular tissue as well, just like with the other photos. So, of course, it has caused damage to the avocado industry. And as I mentioned before, to the backyard avocados. Here, there's a high population of Hispanics, and we all have an avocado and a mango in the backyard. And a lot of people have lost their avocados. And these urban avocados have been really important in the epidemics and in the outbreak because they are serving as bridges in between orchards. So because we are so mixed, urbanized agricultural land, you have avocado orchard, and then you have neighbors, you know, across the street. So these beetles can fly up to 2 kilometers. So you can see, I have seen it with my eyes, and I have monitored a tree in a backyard being infected, and then 2 weeks after, I could see across the street the orchard being infected with the beetles and the disease. So it has played a very important role in the epidemics. So what is going on? The beetle makes these galleries and introduce the fungus to grow the fungus for eating, for nutrition. And while it does that, inadvertently, it introduced the fungus into the xylem. And then because the galleries will cross the vascular tissue and create wounds, then the fungus escapes into the xylem, right? And what happens is that the pathogen induces a defense mechanism that is quite widespread in hardwoods and is very important for hardwoods to avoid decay. And so this mechanism is called tylosis formation. And what it does is that it induces the evacuation of the layer of the vascular system to block and prevent the fungus to spread. But in native Lauraceae, so in American Lauraceae, there is a delay in this response. And so when the tree finally responds, it's too late. The fungus is everywhere, and then it ends up choking itself. And then not only the fungus cannot move, but also the water can move. So it chokes itself. So it's kind of like a hyperimmune response towards the pathogen. And so as the disease progresses, as the days pass, you will see drought stress symptoms. And so it starts with the wilting, then it dries out, and then the complete canopy dries out and dies. So this is a very fast disease. You can lose a big avocado tree in 2-3 weeks, which is, very aggressive. And here you can see how the xylem is being blocked and clogged because of this tylosis reaction. In avocado orchards, what happens is that you have any of these other beetles carrying the fungus and infecting one tree. And because trees are connected below ground in these kind of orchards with big trees and rocky soil, so that the roots don't go too deep, and so they go like on the side, and they are connected, right? And so you can see the spread pattern. I'm sorry I took the video the other way. I should have started with the older ones. But you can see how the disease has moved from tree to tree along the row. And you can lose 100 trees in 6 months if you don't do anything. To let you know, even though we are not Hass producers, California are the ones that produce, California growers are the ones producing Hass. Hass is also susceptible to the disease. So Hass is like 95, 90, 98% of the world commercial production and is susceptible. Also, some of the rootstocks that are used to prevent Phytophthora infection or high salinity or drought impact, those rootstocks that are used in the industry are also susceptible. The current distribution of Laurel Wilt, here you can see at the left in green are the countries that produce avocado. Countries, right? Alaska does not have avocado, but this is like the countries. And then you can see that only in Florida and Myanmar are the production areas that have formally documented this disease. However, the beetle and the fungus is native from Southeast Asia. And so there's multiple countries that have this beetle and fungus in their natural areas. So as I said before, production is increasing. There's more interest in countries cultivating avocado, and so including Asia. And so I really believe that as the susceptible host population increases, so as they plant more American Lauraceae, including avocado, there will be more option for the populations of the beetles to explode and more chances for more introductions into America countries. So far, we know that there was only one introduction in 2002 from Taiwan. And so far, there's no variation in the pathogen population in the US. So we do have genomic data that shows that, but I'm always alert that the pathogen is starting to mutate and vary and create genetic variation. So I'm always, collecting samples to kind of get the genetics of it. The disease was reported in 2004, and this is very important to make to highlight this, that it took 2 years for us to realize that the beetle had invaded the area, because we saw the symptoms of redbay dying. So it took us 2 years. So just think about, right? We have a lot of resources. We have a good surveillance system. Think about the Latin American countries. Think about the South American countries. This is when it gets there, they may not know for a while, and then it will be too late to apply any eradication strategies. That happened to us, actually. So why it has spread so much? Right now it's present in 12 states. And as you can see in this GIF, how it really moved fast throughout the counties. And you see those jumps from not contiguous counties, and then you see that jump into other states. That's because people move firewood. And we all have seen this Don't Move Firewood campaign. It's always, very good campaign, but still people like the smell of lauriceae and they like to take it to their barbecues. And so don't do that, obviously. So that's how the disease has really moved faster than we predicted. And of course, it has also moved through the natural corridors. Persea, borbonia, redbay, and sassafras they have really wide distributions, so that has helped also the disease move widely. But there is more territory to invade, so we still need to be putting resources and effort in understanding this disease and trying to find solutions. Here you can see the counties that have reported the presence of lauriceae, and in red, the ones that already have Laurel Wilt. So there's still a bunch of forest to save and to prevent this disease from getting into. Most of it is sassafras in the east side of the country. In the west side of the country, there is only one native lauriceae, bay laurel, or Umbelaria californica. The distribution is kind of patchy, but there are some contiguous patches of these lauriceae. And so we are very worried about this, right? So the good thing is that there are geographic barriers. There's no hosts in between, and the conditions are harsh to cross that Rocky Mountains. But we are worried that it's going to get into the avocado industry, which is 99% of the avocado production for US. But, this beetle can move by itself, can hijack some of these trade transportation. And we have precedent, right? Citrus greening was first reported in Florida and then in California. So it did move from one side of the country to the other, from one coast to the other one. And even more, we don't have native citrus, really. And so we don't really need native corridors for pathogens and pests to move around. There is a real risk of Mexico getting invaded by the beetle and the pathogen through Texas. The latest report is the presence of Laurel Wilt in Houston area. So it's getting southern and southern. There has been some studies based on the redbay ambrosia beetle on the suitability of this beetle in Mexico, and it's quite suitable for its establishment and reproduction. And this study is only based on the redbay ambrosia beetle. How about the other beetles, which are already there? That's the scary part. There was another study recent that found that native Lauraceae in Mexico from the genus Ocotea is also very attractive to the redbay ambrosia beetle. So not only the environment is suitable for the beetle to establish, but the vegetation is inviting to this beetle. And my lab is working with Mexican collaborators to identify the already fungi present in the area. So they do have wilting avocados, but not because of Laurel wilt, because of other beetles' mass attack to these trees. And these beetles are carrying relatives of the pathogen, but not the pathogen. But that just tells you that it's very easy for these beetles to adopt this new nutrient symbiont into their own fungal community, just like what happened in Florida. So the risk is really layering up into a really bad situation. Of course, it doesn't have to come through Texas. It can come through the trade. And we all heard about these wooden pallets and the danger that they put to our forests. And so when we got the redbay ambrosia beetle in Georgia, it was in 2002. So that was before this certification of wooden pallets. It's called ISPM 15, and it's certified that these pallets are free of wood-boring beetles, right? That's great news. But I have seen with my eyes pallets that have been broken and then they replace the wooden parts with wood that is not under that certification. So that's very scary. Also, you know, when there is a rule, there's also a cheater. And so I can see how you know this can it's not like a really gold standard that everybody's gonna follow. It's good that there is this certification, and I think that is gonna help a lot. But still, we shouldn't be relaxed in terms of surveillance. And the pathogen does not move through the fruit trade. That's a very important thing. So our worry is also from the social part, you know, the social component of it. Mexico's economy is really based and relies a lot on avocado industry, and so as other countries, right? Like Colombia, Chile, Peru, Guatemala is pushing for growing their avocado industry. And so a lot of these economies are really based on or highly influenced by avocado production. So it could be a humanitarian crisis. Also, Lauraceae are very diverse in the Americas, and they are a very important component of ecosystems, right? I was recently in Guatemala giving some talks to avocado growers, and somebody brought up that the quetzal, which is the emblematic bird of Guatemala, only feeds on a specific Lauraceae. So, you know, obviously we know about this with the butterflies and other keystone species or charismatic species that getting, when the host gets eliminated of the ecosystem, they also get eliminated. And so finally, I'm gonna quickly tell you what we are doing. We are working tireless to try to find solutions. We are a transdisciplinary group of people here at TREC, and I am the responsible of the pathology section of the projects. We have plant physiologists, we have horticulture people working on testing different grafts and rootstock behaviors to see if we can find a combination that will slow down the spread of the disease, especially when it's root to root transmitted. We have the entomologists working on pest control, on controlling these beetles through different ways. We have the economics group. Of course, everything needs to be feasible for the growers to apply. And we have a big extension component to it. We work really hand to hand with the growers locally, and also we teach and educate growers in California and abroad. And so laurel wilt management is complicated. It's a very complex disease. Think about citrus greening. It's kind of the same idea. You have a woody perennial host. You have a pathogen that can reproduce and multiply within the host. You have different way of transmission. You have an insect involved in the transmission. So it's a very complicated scenario. And we are doing our best. We have tried different things. A lot of them are grower driven. So they have come up with these ideas, including stamp solarization. You can see here, looks like a ghost, this stamp. So we try a lot of things. A lot of them did not work. So far, we can only recommend sanitation and tree disposal. You have to remove the tree with roots and everything. So really take it out. We also recommend to remove the adjacent trees, even if these are asymptomatic or not showing symptoms, which is something that the growers don't like to hear. Nobody wants to remove a healthy looking tree. We also recommend to increase light. So prune really good the canopy. This is good for hurricanes too, but also it's good to increase the light because these beetles like kind of like a forest type of ecosystems with a lot of shade. So if you increase the light, then you will decrease the beetle population and reduce tree stress. So even though the redbay ambrosia beetle seems to affect healthy trees, the other beetles are more attracted to ethanol and to stress signals produced by stress trees. So again, this is not a practice that people like to hear, but that's the recommendation that we have. And they say that a picture tells you more than 1,000 words. So you can see here, my grower, he is a volunteer in my lab. He has a strict sanitation program and scouting. And you can see the neighbor using all sorts of, strategies like injections and solarization and stuff like that. And you can see the difference, right? He is doing well. He is mitigating the effect of the disease, but some other growers have lost their entire orchards. So, okay, sorry, that was a duplicate. So my lab, again, I'm in charge of the pathology section. So I'm doing a lot of things, following different lines of research, always asking for money. This is expensive research. We need a lot of labor and a lot of resources. So I'm doing germplasm screening. So I made a collaboration with the largest germplasm of avocado in the world, located in Mexico. It's called Fundación Sánchez Colín. And they are sending me seeds from non-commercial varieties. Some of them are almost non-domesticated. So really unique, really amazing germplasm, land races, that's what they call it, land races. And so I'm doing a lot of this screening. I'm also testing different fungicides that hopefully give the growers a first line of defense. I understand this is not sustainable, but we need something now. We can work towards finding more sustainable solutions, more long-term solutions, but for now we need something fast. Then I'm also improving the detection techniques and the risk analysis for places or production areas where the disease is not there yet. So I'm working with several countries and teaching their diagnostic personnel, their surveillance personnel, how to identify the disease. And I'm also working on biological control and trying to find natural enemies that can mitigate the effect of this. So we are a very busy lab. We are always planting or killing. These are like the two spectrums of my research. We have to plant a lot and then we inoculate a lot, and hopefully we get either survival because of the strategy that we are using or because of the genetics of that material. We also do a lot of lab work. We are trying to find metabolites from Trichoderma species that we can use to kill the fungus, and we are doing some progress. It's a long road. and a painful actually to walk, but it's very exciting. So you can read everything about the tolerance that we have found in this germplasm from Mexico. Now, I have to say that these tolerance that we have, seemingly tolerance that we have found in this material, these are greenhouse studies. So then we have to put in the field, let it grow, replicate the experiment with clonal material, and see if the tolerance that we are observing in the greenhouse translates in the field. So this is basically a breeding program for a perennial. So it's really heavy, heavy stuff. But you can go to that publication and learn more about it. We also found a new compound, a conventional pesticide that seems to have amazing mobility within the vascular system of the tree. So it's very exciting and working with the companies. And now we are going to move the trials into the field this season. So I'm super excited about it, and really growers need something, and I'm very hopeful of this chemical. And we are also finding antagonistic trichoderma that can become endophytic. So basically, I'm trying to find natural enemies that I can inoculate into the seeds of avocados, and when the avocado germinates, this fungus, the good fungus, colonizes systemically the plant, and then it will be able to fight the bad fungus once it gets infected. We'll see. I mean, we have some mixed results. Interesting, but still we are far away from finding something that is sustainable. And with that, thank you for listening. Again, my lab works on several aspects of plant health. We have the plant disease clinic. We also take samples from nature. It's a little bit difficult to pinpoint the problem in plants coming from nature, but we are here to provide a service. So just let us know if you have any questions. Thank you.
I turned my sound up for everyone else. That was great. We had one question sent in ahead of time by one of our members, Mike Koner. I don't know if he's on just yet. He says, please ask if she can include information on the related lancewood, Demburnia coriacea, which is a native tree. And I looked it up, and I'm not familiar with this plant, and it's in coastal East Coast Florida, and then down to Southeast Florida. And it looks like maybe a disjunct population in Highlands County.
So it is a lauraceae? I'm sorry, I'm not familiar.
It's a lauraceae.
Yeah. So if it's a native lauraceae, it's very likely susceptible. I can look up if it has been tested. I have been here for 7 years, so I took over my predecessor program, Randy Plots, and they did work with native lauraceae testing the susceptibility of the species. And so I can look up to see if it has been tested. And if it hasn't been tested, and there is a possibility of getting seeds to do the experiments inside the greenhouse, I will be very interested in testing the susceptibility of this group. Mm-hmm.
Cool. Also, he says, Carl Terwilliger of Meadow Beauty and longtime respected grower suggested that dispersed planting of the genus, maybe he's referring to genus Demburnia, may keep it viable as the wilt decimates crops in wild areas. I would like to know if there's any validity to this thought, and whether planting across developed areas will create linkage and help it spread.
Yeah, so that's a good point, right? Connectivity of susceptible hosts is very important for epidemics, and having isolated plantings could be a strategy. So let's say it could be a strategy maybe for California or for other countries where the production area of avocado is not neighboring forest, which that's what happened in Florida, right? We had this mix of patches of redbay and swampbay very interconnected with the orchards. And we have the backyard avocado. So it was kind of recipe for disaster, right? So definitely isolation of production of productive orchards could be a way. I don't know how would you do that with the forest, because you also don't want to fragment forest, because connectivity is important for other players in the food chain. But that's something to think about, right? I've been thinking about, and that's not a new idea, but I've been thinking about surrounding the avocado orchards with pines, which is not a host for the fungus. And so I have seen in Central Florida few avocado orchards surrounded by, not Central Florida, like Fort Pierce, around that area, surrounded by pines, and they haven't got infested with the beetle. So I think this strategy may work. So that's something to, but again, you have to wait until the pines are big enough to be a barrier for flight for the beetles. And then they have to go into the pine. And then once they go into the pine, the fungus will not establish. So the symbiont of the next generation will be a different fungus, not the pathogen.
So are they going into the pines? They're going in and eating they don't eat the wood. Are they
They don't
eating
eat the wood.
Are they eating a different pathogen in the pine?
Yeah, so ambrosia beetles don't have only one symbiont. They have a preferred symbiont, like the redbay ambrosia beetle. Mostly it's pocket community, like the Macandria, which is the structure where they store the fungus. Has many species, but is dominantly Harringtonia lauricola, the pathogen. But the other beetles, they have a combination of Harringtonias and Raffaeleas, and they have a more diverse community. And so my hypothesis is that when they encounter a host that is reproductively suitable for the beetle, they culture a different fungus. And so then the pathogen will lose abundance, and then the community turns over. So when the female leaves the pine, it may be living with other more abundant fungus than the one that is causing disease. So again, these are very difficult hypotheses to test, but I think they're worth to look into.
And so before we went live, we were discussing the implications of your research for native members of the Lauriceae family, and you were saying that while you haven't tested your method specifically on native members of Lauriceae, these would be applicable.
Correct. So this is the same fungus. So it's the same pathogen. So whatever works in avocado or in the lab, it should work in the forest, right? So if I'm finding a pesticide that kills the fungus, it should kill the fungus in the forest species as well. They might have a different absorption rate, different vasculature, but I would say that pretty much it will be applicable. Now, of course, you cannot use pesticides in the forest, but for homeowners that perhaps have a very valuable tree, for germplasm collections, for botanical gardens, places like that, they could apply some of these more production type of strategies. But yeah, so I think that whatever we do, it can be work in the other species. The germplasm screening, it's important because I'm not really trying to replace Hass. Hass will never be replaced. That's the main cultivar. But by finding tolerant material, we can understand the mechanism of tolerance. And if that mechanism is tied to the genetics, eventually we can find the regions that are responsible for that, and then we can manipulate the genomes of plants, right? So like the chestnut right? The American chestnut. Now there's a program for introducing a genetically modified plant, and there is a controversy. I understand that, but we are just looking at the science part of it.
We have a question from Steve Woodmansey. He asks, at what size DBH does the pathogen begin infecting trees?
That's a very good question. So the pathogen will infect trees at no matter what diameter. I can infect baby plants by artificially injecting the plants. But the beetle, since the beetle needs to create a gallery, is not gonna look for trees. And I don't have exactly the number. I'm just trying to look for my ruler because I have problems with inches and centimeters. But I would say, I don't know, 5 centimeters, 9 centimeters. So that's very important because people are replanting avocado. And so now they are baby avocado. So they are not attractive to the beetles. So we are buying time in trying to find a solution for when these plants become attractive to the beetles. So let me see, I have a ruler here. I would say 10 centimeters, actually. That's probably the diameter that they will go for to build a gallery. But I can artificially inoculate plants with 1 centimeter diameter and they will die. So it's not about the fungus is able to infect anything, but the beetle is the one that will introduce the fungus into the tree.
Then we do have a question about avocado cultivation.
Cool.
Someone is asking, Patty Pritz is asking, what is causing a 6-year-old avocado to drop all the fruit when it is about the size of a pea? She says it is a patio species day in a 28-inch pot.
That's, without really context, it's difficult to say, but avocados don't like pots, number one. They really do very bad when you have them in containers. And so I think that it's not really supporting the trees because it just cannot provide for those fruits. Sorry, it's aborting the fruits because it cannot provide for those fruits. So once the fruit is not gonna really grow, it just drops it. And I think it's likely, I don't know where this person say where they are. Okay, so, but I know for sure that avocados do not like containers. They get root bounded and then they get nutrient deficiencies. And, I can see why they wouldn't invest in production, right? They just drop the fruits.
I have a question about the life cycle of the fungus once it has infected the tree. The tree is essentially strangling itself in effect in a way to prevent the spread of the fungus. But I have seen some bay trees resprouting after that looks like they've died. So are the root systems completely dead? Are the plants continuing to be infected with the fungus? And what is like the long-term outcome once a plant has been infected?
Yeah. So you're correct. So the tree chokes itself. And so that's when you get the drought symptoms because water can no longer be uptaken. But Lauraceae, and I think I have seen this in sassafras, in redbays, in avocado, they resprout. The pathogen colonizes the roots for sure. I mean, that's proven. And very quickly. In artificial inoculations, 10 days after I inoculate, it's already in the roots. But we have seen many avocados resprout. That's just their nature to resprout. And young tissue is more tolerant to the pathogen. So I don't know the mechanisms, but when I inoculate small plants, I have to put 1 million spores, 1 million spores. But when I kill a tree in the field, I only need 300. So if I put 300 in the baby plant, it doesn't die. I think it's because they have a simple vascular system that is not connected on the sides. So it's like a tube. So the pathogen can move up and down, but not laterally. So there are some vessels that are still functional. And then, because we are in the tropics, the plants can create another layer of xylem quite quickly. And we have seen that in the greenhouse. And so when a lauraceae resprouts, the resprout, it's equivalent of young vascular tissue. So you don't see them sick until the fungus reproduces and the vascular system starts connecting and becoming more complex, and then the fungus will spread more widely. And so we have tested this hypothesis, proven it in resprouting avocados, and it's super interesting, and it's amazing how it can really be a problem for management, right? Because when I went to Tennessee, the forest manager, the park ranger, was telling me how they have stumped the sassafras and they have, mitigate the problem. And then I'm like, So I opened the tree and I opened the baby resprouts, the saplings, the water sprouts, I think they're called, and then they were infected. So from the management point of view, you should remove that section, right? And so what's the long term? I have some data that shows that those resprouts eventually will collapse.
Yeah.
If the fungus is there, they will collapse. Now, that because of multiple variables, in one occasion, the fungus was able to efficiently be walled off, and that resprout survives, that is very low incidence. That's just by chance of all these environmental factors working together. And so that fungus just remained there, wall off, right? So that could happen. I'm not saying that not, but that's very confusing for growers, right? And for park rangers, because if they say, no, but they have survived. So which is it, right? And I have to explain to them that there is the chance variable playing sometimes, right? And so, but the fate in general is that those resprouts will die once they get to the diameter that allows the fungus to move more widely.
Okay. And if they were to remove the dead tree and replant, whatever is replanted, it's not going to spread through the soil. Is that correct?
That's correct. So it should not survive in the soil. And I have a colleague that actually the avocado grower found a root that had been cut from, like whenever they removed the tree, they left a piece of root buried for like a year. And then, but it was still moist when we recovered it. And then we were able to plate the fungus out of it. So as long as there is tissue that has some moist, the fungus will survive. But once it dries out, it just dies. So, and that piece of root is not going to form any connection with a new plant, right? So it's safe to replant in Laurel Wilt infected areas.
Okay, thank you.
Awesome. That's all the questions that we have. Patty did get back to us and said that the nursery that sold it to her touted it as a potted avocado.
I never heard about potted avocados that can produce fruit. But maybe, I mean, I don't know which variety it was. Is it day variety? I'll look into it. I never heard about it, but I'm not a horticulturist, so.
Yeah. So your talk was excellent. Thank you so much for joining us and for all of your work on this kind of scary pathogen that we've been dealing with for quite a while now.
Yeah. Thank you for having me. Again, I am really keen on nature, so hopefully I can do more research on other ecosystems that hopefully they don't have any problems, so I don't have to work on them, but I'm here if something happens.
Thank you.
Okay.
And
Good.
so just letting all of our Lunch and Learn viewers know that we have a Lunch and Learn scheduled. The policy and legislation, how to get involved in policy, that was supposed to be in the end of September, will be now at the end of November, but we're going to skip Thanksgiving week, so it'll be the 22nd of November, that Friday before the last Friday. So it'll be still the fourth Friday at noon Eastern, and we'll have Jean Kelly, our policy and legislation chair, as well as our lobbyist, Travis Moore, and then staff, me, probably Lily, perhaps our new executive director, Melissa. So please join us then for an invigorating discussion about how you can get more involved in policy at your local level and state level with FNPS, and just a quick overview of everything that happened with the state parks issue, and maybe an update on the Withlacoochee land swap if we have one. So with that, thank you, Marina. It was great working with you. Thanks, Lily, for the questions coming on.
Thank you, guys. Nice to meet you.
And I hope everyone has a great weekend.